Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Ever get the feeling that when you're under the weather, you just kind of want to be left alone? You're not really up for socializing.
Right, definitely.
Well, what if I told you that there's some fascinating new research suggesting that the same signals your body uses to fight off a cold might actually be playing a role in how social you feel?
Yeah, it's pretty wild, isn't it?
Totally. So today, we're going to be taking a deep dive into a study that did this incredible job of mapping out where these immune system receptors are located throughout the brain. And get this, they found that one specific signaling molecule seems to be super important in how we connect with other people.
I mean, we're learning more and more about just how interconnected different systems in our bodies really are.
For sure.
For a long time, we thought about the immune system and the nervous system as these totally separate things, but the research is really showing us that's not the case at all.
Yeah, it's so much more complex than that.
They're constantly in communication, back and forth, and they're influencing each other in really profound ways.
Right. And that's exactly what this deep dive is all about. We're going to be exploring how specific parts of the immune system, these signaling molecules called interleukin 17, and the receptors they bind to in the brain, seem to be involved in social behavior.
Yeah, definitely.
We'll be focusing on a new scientific paper that's uncovered this really specific interaction happening in the cortex, which is that outer layer of the brain.
Right.
And to help us make sense of all of this, we also have some insights from a related health news report to give us a little bit more context.
Awesome.
So what we really want to do today is break down the core findings of this research and try to understand what it all means for how social behavior works at a biological level.
Yeah, exactly.
We'll also touch on some of the unanswered questions that this research brings up because, like with any good scientific study, there's always more to explore.
Oh, absolutely. There always is.
Our goal is to give you a clear, engaging explanation of this pretty complex science so that you come away feeling like you really learned something, but not overwhelmed by all the details.
Right. We want you to walk away feeling like, okay, I get that.
Exactly. All right, so let's start with this study itself. What were the researchers hoping to understand with this particular piece of work?
So their main goal was to investigate how these interleukin 17 cytokines, which are these signaling molecules, and their receptors were distributed throughout the brain.
Okay.
They were particularly interested in whether and how these molecules might be influencing social behavior. There had been some earlier studies that hinted at a possible connection, but the specifics were really unclear.
So they had this kind of general idea that there might be a link, but they didn't know exactly what it looked like or how it worked.
Exactly. So this study was all about trying to fill in those gaps and get a clearer picture.
Okay, so how do they go about mapping these receptor proteins across the entire brain? I mean, that seems like a really massive undertaking.
It really was.
Super detailed.
Oh, yeah. They actually created these incredibly detailed maps that show exactly where all the different subunits of the interleukin 17 receptors are located in the brains of mice. And to do this, they used genetically modified mice where these receptor subunits basically had a little tag attached to them.
Okay, like a little flag.
Exactly, like a little flag. So the researchers could see where those receptors were. So imagine, if you had this really intricate map and you put these little flags on all the specific locations you wanted to highlight, you could then step back and see the bigger picture.
That's a great analogy.
That's essentially what they did here, but at a molecular level.
Cool. So they had their map.
Yep.
And then they also decided to zoom in on a particular area of the brain. Can you tell us about that?
Right. So they focused on a specific region within the cortex called the somatosensory cortex dysgranular zone, or S1DZ for short. They chose this area because earlier research had suggested that it might be connected to problems with social interaction and repetitive behaviors, which kind of made it a prime spot to look for potential roles for these immune signaling molecules.
So they're kind of like, all right, we have our map. Let's focus in on this region that we think might be particularly interesting.
Exactly.
So they have their map. They have their area of interest. What did they do next to try and figure out what these receptors actually do in relation to social behavior?
So to really get at the function of these receptors, they used some pretty sophisticated genetic engineering techniques in mice. They created what are called knockout mice. where they specifically removed certain interleukin-17 receptor subunits, like interleukin-17 receptor B and interleukin-17 receptor C.
Oh, so they're just getting rid of them entirely.
Yeah, they're taking them right out of the equation in these specific mice. And then they also did the opposite. They created mice where those same subunits were overexpressed, meaning there were way more of them than usual in certain brain regions. By observing how the behavior of these genetically altered mice differed from regular mice, they could start to piece together the roles of these receptors in social behavior.
So it's like they're turning the dial up or down on these receptors and seeing what happens.
Totally. It's a really direct way to test how important these receptors are.
Okay, I see. But I'm also seeing that they used another method where they actually gave the mice the signaling molecules themselves. How did that work?
Right. So in addition to manipulating the receptors, they also wanted to see what would happen if they introduced the signals that those receptors bind to.
Okay.
So they used this non-invasive method to deliver interleukin-17A and interleukin-17E directly into the brains of the mice.
And they did this through the nose.
Yeah, through the nose.
That seems a little strange.
I know, it sounds a bit odd, right? But it's actually a way to bypass the blood-brain barrier, which is this protective layer around the brain that makes it hard for certain substances to get in.
Oh, interesting. So they're basically finding a way to sneak those molecules into the brain.
Exactly. It's like a secret back entrance.
Mm-hmm.
And this allowed them to see how introducing those signaling molecules themselves would impact behavior.
Makes sense. So they're manipulating the receptors, they're introducing the signals, and then, of course, they needed a way to measure whether any of this was actually changing social behavior.
Right, exactly.
Right.
So they put the mice through a bunch of behavioral tests that were designed to assess their social skills.
Okay.
Things like how much time a mouse spent interacting with another mouse, how they reacted to social cues, that sort of thing.
Yeah.
And by comparing the mice with altered receptors or those that guide the signaling molecules to regular mice, they could start to figure out if the interleukin-17 pathway was having any real impact on social behavior.
It's a really elegant experimental setup when you think about it.
It is, yeah.
They're really thinking this through from multiple angles.
Definitely.
So the last piece of the puzzle here seems to be this technique called translating ribosome affinity purification analysis, or TRA. What is that? It sounds pretty technical.
It is a bit technical, but essentially it's a way for scientists to zoom in and see what's happening at a molecular level within specific cells.
Okay.
They used it to identify the specific genes that were being expressed or turned on in neurons that had different types of interleukin-17 receptors. Ribosomes are like these little factories inside cells that build proteins, and by looking at the messenger RNA molecules attached to ribosomes, they could basically get a snapshot of which genes were active in these neurons.
So they're looking at what's happening inside the cells themselves.
Yeah, they're going really deep into the molecular machinery to understand what's going on.
Wow, that's pretty impressive. Okay, so they used all of these different approaches to investigate this question.
Right.
What were some of the key things they found out? What are the big takeaways from this study?
Okay, so one of the first big findings was that the different subunits that make up the interleukin-17 receptors, they actually have really unique and distinct patterns of expression throughout the brain.
Interesting.
Yeah, so it wasn't just like they were all evenly distributed everywhere. For example, they found that interleukin-17 receptor B was present in the cortex, including that S1DZ region we talked about.
Okay.
But interleukin-17 receptor C wasn't as prominent in those deeper layers of the cortex.
So different receptors are showing up in different parts of the brain. It's not just this uniform distribution.
Right, exactly. And that suggests that these different receptor subunits might have different roles in different parts of the brain. It's like they each have their own special job to do.
Makes sense. So what else did they find?
Okay, so here's where it gets really interesting. They discovered that it's interleukin-17 receptor B, not interleukin-17 receptor C. That seems to be the key player in how interleukin-17A affects social behavior.
Okay, that's interesting.
They use this mouse model that mimics certain aspects of neurodevelopmental disorders. It's called the maternal immune activation model. And in these mice, which often have social deficits, treatment with interleukin-17A usually improves their social interaction. But when they got rid of interleukin-17 receptor B in these mice, the interleukin-17A didn't work anymore.
So it's like interleukin-17A needs to interact with that specific receptor, interleukin-17 receptor B, to actually have that positive effect on social behavior.
Exactly. It's like a key fitting into a lock. If you have the wrong key, it's not going to open the door.
I like that analogy. So they're really starting to pinpoint the specific molecules and receptors that are important in this pathway.
Right. And then they also found something else really fascinating. They identified another signaling molecule, interleukin 17E, which is also known as interleukin 25, as being super important in this whole process.
Okay, so we have another player entering the scene now.
Right. And they found that this interleukin 17E is actually produced by neurons in the cortex itself.
Wow, so the brain is making its own immune-like molecules.
It is. That's a huge finding. And this interleukin 17E seems to boost social interaction by acting on neurons that have both interleukin 17 receptor A and interleukin 17 receptor B.
So it needs both of those receptors to work.
It seems like it.
Okay, interesting.
Yeah, so it's not just the immune system sending signals to the brain, but the brain is actually talking back using a similar language.
That's pretty amazing. It really does show that it's not these two separate systems, but this much more integrated back and forth communication going on.
Yeah, it's a much more dynamic relationship than we used to think.
For sure. So they found this interleukin 17E. It's made by the brain. It seems to be important for social behavior. What else did they discover?
Well, they didn't stop there. They also found that when they treated mice with interleukin 17A, it actually increased the levels of interleukin 17E in the cortex. So that suggests that there's this sort of chain reaction happening where one signaling molecule is promoting the production of another.
Wow. It's like a domino effect almost.
It is, yeah.
So how does this interleukin 17E actually influence the neurons to affect social behavior? What's the mechanism there?
So using some super precise electrical recordings from individual neurons, they were able to show that interleukin 17E actually directly reduces the activity of neurons in that S1DZ region that express interleukin 17 receptor B.
Okay, so it's calming things down.
It seems that way.
Interesting.
Like it's turning down the volume on those neurons.
Okay, I like that analogy.
And that makes sense because if you think about it, maybe excessive activity in that particular brain region is somehow interfering with normal social interaction. So by calming those neurons down, interleukin 17E might actually be helping to regulate those social behaviors.
Okay, so it's not just about sending signals, but it's about fine-tuning the activity of those neurons.
Right, exactly. And then finally, they found that some neurons in the S1DZ actually have both interleukin 17 receptor A and interleukin 17 receptor B on their surface. And that's important because interleukin 17E seems to need to bind to both of those receptors to have its effect. They confirmed this by selectively removing interleukin 17 receptor B from those neurons, and that actually blocked the social enhancing effects of interleukin 17A. So that really strengthens the idea that this specific pairing of receptors is super important for this pathway.
It's like a double lock that needs both keys to open.
It is, yeah.
Okay, so they uncovered this really detailed sequence of events showing how these specific molecules and receptors in a particular part of the brain are really important for social behavior, at least in these mouse models.
Right.
So let's shift gears a bit and talk about what all of this means. What are some of the implications of these findings for our understanding of how our brains and our immune systems interact?
I think one of the most significant implications is that it really underscores how these immune molecules can act as neuromodulators. We tend to think of cytokines as being primarily involved in the immune system, right? Like fighting off infections, dealing with inflammation. But this study, along with a growing body of research, is really showing us that these same molecules can also directly influence how our brains work and ultimately how we behave.
So it's not just that the immune system keeps us physically healthy, but it's also playing a role in how our brains function.
Exactly. It's breaking down that traditional separation between the immune system and the nervous system.
Yeah, it's blurring those lines.
It's all much more interconnected than we thought.
For sure. And does this understanding open up any new possibilities for therapies or treatments?
It does, actually. The fact that they pinpointed this interleukin 17E and interleukin 17 receptor B pathway in the brain as being involved in social behavior suggests that it could be a potential target for developing new treatments for people who have social difficulties, especially those with neurodevelopmental disorders.
That's a really hopeful idea, especially because these kinds of social challenges can be really difficult to treat.
They can be, yeah. And the researchers actually found that the human regions of interleukin 17A and interleukin 17E had similar effects in mice, which makes this possibility even more promising. Still early days, of course, but it's a really exciting avenue for future research.
It definitely is. And it also highlights how important it is to continue studying these intricate connections between our immune systems and our brain.
Oh, absolutely.
There's so much more to learn.
This study gives us this really valuable framework for understanding how the immune system and the nervous system are communicating with each other to influence these complex behaviors like social interaction. And by continuing to map where these cytokine receptors are in the brain, we might uncover other surprising roles for immune molecules in different brain circuits.
And potentially in other behaviors as well, right?
Absolutely. Who knows what else we might find?
That's so cool. Okay, so as with any scientific study, it's important to consider the limitations. What were some of the things that the researchers themselves acknowledged about their work?
Yeah, for sure. So one limitation is that they used a technique where they could see the cells that were expressing the receptors, but the intensity of that signal might not have perfectly matched the actual levels of the receptors in the brain.
Okay, so it's like a good indicator, but not necessarily a precise measurement.
Yeah, exactly. They also noted that for one of the receptors, interleukin 17 receptor C, their results might have included some data from cells that weren't actually neurons. So the information about its expression might not be completely specific to neurons. It's possible there's a bit of background noise in that data.
Okay, so a little bit of a caveat there.
Right. Another limitation was that the viruses they used to deliver genetic material into the brain, they might not have been equally effective in all types of brain cells, especially microglia, which are these important immune cells in the brain. So their analysis might have been more focused on neurons than on those other cell types.
Got it. So there are some limitations in terms of which cell types they were able to really look at closely.
Right.
Okay. Anything else?
And finally, the study mostly focused on social behavior in the context of this maternal immune activation model, which, as we talked about, is useful for understanding neurodevelopmental disorders. But they acknowledge that more research is needed to see if interleukin 17B and interleukin 17E play similar roles in other types of behavioral issues related to maternal immune activation, or even in typical, everyday social behavior.
Okay, so they're being appropriately cautious and saying, look, we found some interesting things, but we need to do more research to see how broadly these findings apply.
Yeah, exactly.
Okay, that makes sense. Now, we also took a look at some expert opinions about this research. What were some of the overall perspectives that other scientists in the field had on this study?
I think a lot of the experts were really impressed by how this study further solidified the idea that the immune system's influence goes way beyond just fighting infections. It's clearly involved in shaping brain development and behavior, which is a big deal. It's really changing how we think about these systems.
So they're seeing this study as further evidence that the immune system is playing a much bigger role in the brain than we initially thought.
Absolutely. And by identifying where these immune receptors are located and what they do in the brain, this research opens up so many exciting possibilities for future studies on neuroimmune interactions, both in healthy brains and in brains affected by neurological or psychiatric conditions.
So it's not just about understanding the immune system on its own, but understanding how it interacts with the brain in both health and disease.
Exactly.
That's a really important point. And the discovery of interleukin 17E as this neuromodulator that acts on these specific groups of neurons in the cortex, that was also seen as a really big step forward in understanding the biology of social behavior.
Yeah, that was a big one.
But of course, there are always more questions to be answered.
Yeah, oh, always, always. And one of the things that the experts highlighted was the need for more research to really understand how interleukin 17A controls the production of interleukin 17E in the brain.
So they're saying we need to understand that upstream connection better.
Right, like what's triggering that initial step in the pathway.
Okay, so more to be explored there.
Definitely.
All right, well, as we wrap up this deep dive, we always like to leave you with something to think about.
Right.
So this research on the relationship between the immune system and social behavior really makes you wonder what other seemingly unrelated systems in our bodies might be subtly but significantly influencing our thoughts, feelings, and behaviors in ways we don't even realize yet.
That's a great question.
It's kind of mind-blowing, right?
Totally. This study is a perfect example of how basic scientific research can uncover these totally unexpected connections.
Exactly.
If you're curious to learn more about neuroimmune interactions or how cytokines work in the brain, I encourage you to dive into the scientific literature.
Yeah, there's so much out there to explore.
Just searching for terms like neuroimmunology and cytokines in the brain will give you a great starting point.
And here's another thought. Environmental factors that can affect the immune system, like stress or infection, could be indirectly impacting our social interactions in ways we're only just beginning to understand.
Yeah.
So could this research lead to new ways to help people who have social difficulties?
It's definitely a possibility, and it's a really important area for future research.
It is. There's so much potential there. Well, that's it for this deep dive.
Thanks for listening.
Thanks for joining us.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.